For over a century, farmers and agricultural scientists have basically been staring at this massive mystery. It plays out in fields all over the world every single day.
Yeah, it's a very practical problem.
Right. You take a perfectly healthy crop, you deprive it of a single trace mineral, which is boron, and the results are just, well, they're catastrophic.
Totally catastrophic.
The roots stunt, the growth becomes entirely chaotic, and eventually the meristems, the growing tips of the plant, they just wither and die.
Exactly.
So for 100 years, everyone knew that without boron, a plant literally falls apart. But nobody actually knew the mechanism.
Right. It's one of those fundamental biological puzzles. The macro level effect is incredibly obvious. I mean, entire harvests can fail due to boron deficiency.
Wow, yeah.
But the micro level mechanism, the actual molecular, the why, was just a complete black box.
It's completely unknown.
Yeah. The agricultural world knew boron was essential, but not what it was actually doing on a cellular level to prevent that structural collapse.
And that is our mission for this deep dive, because a recent breakthrough paper finally managed to zoom way, way in, right down to the microscopic space between a plant's cell wall and its membrane.
It's a fascinating study.
It really is. The source we're looking at today is a paper from the journal Cell titled Boron-Bridged GIPCs, Stabilized Cell Wall Anchoring, and Pin Polar Domains.
Which is a mouthful, but we're going to break it down.
Yes, we're going to explore exactly how they solved this 100-year-old mystery.
Well, to really grasp what these researchers found, we have to less rethink the basic architecture of a plant cell.
Right, because, I always picture the plasma membrane just sort of pressing up against the cell wall via turgor pressure, like a water balloon inflating tightly inside a rigid cardboard box.
That's the classic analogy, yeah.
We know the membrane is flexible and holds the fluids, and the wall is rigid to let the plant stand tall. But the assumption was always that they were just pressed together by internal water pressure.
Right, and that assumption is exactly what this source material overturns. These two layers aren't just, sitting next to each other, passively held together by pressure.
They aren't.
No, they are molecularly bolted together. And boron, that mysterious trace element we mentioned, is the physical thread holding the entire structure intact.
Okay, so if boron is the thread, what exactly is it bolting together? I imagine the cell wall is pretty complex, but the plasma membrane is mostly just a fluid layer of lipids, right? Where is the anchor point on that water balloon?
So the anchor points are specific lipids called GIPCs.
GIPCs.
Yeah, which stands for glycosilynositol phosphorylceramides.
Wow. Okay, GIPCs it is.
Right, much easier. Now, these aren't just rare niche molecules floating around. GIPCs are incredibly abundant. They make up anywhere from 25 to 50% of the total lipids in a plant's plasma membrane.
Wait, really? A quarter to half of the membrane surface is practically paved with these specific GIPC molecules.
Exactly. So they are a foundational building block of the plant cell's outer boundary.
So how does boron physically connect to them?
Well, boron acts as the bridge because the borate ion has this unique chemical property. It forms bonds with molecules containing diols.
Diols.
Yeah, a diol is basically just a molecule that has two hydroxyl groups sitting right next to each other. So the researchers wanted to see if boron was directly grabbing onto the sugar heads of these really abundant GIPC lipids.
But how do you actually see that? You can't just put a single lipid under a standard microscope and look for a tiny boron atom hanging off of it.
No, definitely not. They had to use some pretty heavy-duty mass spectrometry, specifically Maldi-Toff and 11B NMR spectroscopy.
Okay, that sounds intense.
It is. They ran this on total GIPCs extracted from Arabidopsis seeds. Conceptually, they essentially vaporized the lipid molecules and weighed them incredibly precisely to see exactly how much boron was, hitching a ride.
Oh, I see. So they're looking for the extra molecular weight of the boron atoms attached to the sugar.
Yes, exactly. And the data they got back was striking.
What did it show?
Well, GIPCs are classified into 6 series, A through F, based on how many sugar residues they have. The mass spec data showed that all 6 series possess multiple boron binding sites.
On all of them?
Yeah, on both pentose and hexose sugars. And series A, which is the most abundant type in the membrane, can actually bind up to 4 boron molecules simultaneously.
4 of them?
Yeah, it's an incredibly dense, multivalent binding hub.
Okay, let's unpack this. If the cell wall is a rigid brick wall and the membrane is a flexible sheet of canvas, the GIPCs are like heavy-duty grommets punched all over that canvas.
That's a great way to visualize it.
And boron is the carabiner, clipping every single one of those grommets directly to the bricks.
Yes, precisely.
But wait, if a plant is growing, it has to flex and stretch.
Mm-hmm.
Is this connection permanent? Like if you clip a carabiner, it's locked. Can the plant unclip it?
What's fascinating here is that the chemistry of these borate ester bonds perfectly suits the dynamic life of a plant.
How so?
These bonds are covalent, meaning they involve sharing electrons, so they are quite strong. But they are also reversible and dynamic. They can swap and exchange under different environmental stimuli, like tiny shifts in pH.
Oh, so it's more like molecular Velcro then.
Yes, exactly like Velcro.
It holds tight under tension, but the plant can sort of rip it apart and stick it back together if it needs to expand or shift.
Right. That dynamic covalent bonding allows the plant to maintain a continuous, powerful physical attachment that is still flexible enough to adapt to growth, movement, and environmental stress.
Okay, so just to do a quick recap here, we've got the wall, the membrane, the GIPC grommets, and the boron Velcro holding them together. We have a clear picture of this now.
We do.
But, in biology, just because two things can bind together in a test tube doesn't mean that binding is actually structural in the living organism.
That is a very important distinction.
Right. So how did the scientists prove this Velcro actually keeps the cell's physical structure from collapsing?
Well, by attempting to break the connection in a living cell. Yeah, they utilized a phenomenon called plasmolysis. If you expose a plant cell to a highly concentrated salt or sugar solution, the water inside the cell rushes out.
Because of osmosis, right.
Exactly. The flexible plasma membrane begins to shrink and pull away from the rigid cell wall.
So you are artificially deflating the water balloon inside the box.
Yes, and when you do this to a normal, healthy plant cell, the membrane doesn't just peel away cleanly.
It doesn't.
No, under a microscope, you see these tiny, thread-like structures stretching tightly between the retreating membrane and the cell wall, fighting to hold on.
Oh, wow.
These are called hessian strands. They are the physical, visual proof of that tethering we've been talking about.
Okay, so they see these microscopic threads snapping under tension. But how do you prove boron is the specific thing holding them? You can't exactly grab a single atom out of a living strand.
Right, so they introduced a chemical cap called PBA, or phenylboronic acid.
Phenylboronic acid.
Yeah,
Okay.
PBA is structurally very similar to natural borate, but with one critical difference. It only has one pair of hydroxyl groups instead of two.
Meaning it only has one hand to grab with.
Exactly. It's a competitive inhibitor. It can bind tightly to the dials on the GIPC grommets, but it has no free hand left to reach across and cross-link to the cell wall.
I see.
It essentially acts like a cap on the sticky end of the GIPC, neutralizing the ability to form a bridge.
That is a brilliant trick. It's like watching a sci-fi tractor beam suddenly switch off. You just cap the projector, and the physical hold completely disappears.
And the visual results were dramatic and incredibly fast. Within 10 minutes of applying that PBA cap, those tension-bearing hessian strands completely vanished.
Just 10 minutes.
Yeah. When the researchers induced plasmolysis this time, the cellular membrane just collapsed perfectly smoothly inside the cell wall. There was zero resistance, no tethering left at all.
A plant cell is incredibly sensitive, though. If you dump a chemical acid onto it, couldn't the cell just be dying?
That's a fair question.
Yeah, like how do we know the PBA didn't just poison the cell and cause the membrane to break down organically?
The researchers anticipated that exact critique, which is why they ran a highly specific control using a milder analog called ETBA.
ETBA.
Yeah, ethyl boronic acid.
Yep.
ETBA has a very unique chemical property. It only becomes active and capable of binding at a highly alkaline pH around 10.5.
Oh, wow. So at a normal, mildly acidic plant pH, it just floats around doing nothing.
Exactly. It's a chemical switch they can turn on and off just by changing the acidity.
That is so clever.
They showed that ETBA only disrupted those microscopic tethers when the pH was artificially raised to 10.5. At normal pH, the tethers held strong.
Which proves the point entirely.
Right. This proved beyond a doubt that the collapse wasn't just general chemical toxicity destroying the cell. It was a highly specific, chemically driven disruption of the boron cross-linking.
Okay, wait. So if boron is basically acting as a molecular carabiner, locking the membrane to the cell wall, why does the plant actually care if it's locked down?
What do you mean?
Like, a water balloon in a box is still protected, even if it's not glued to the sides. Does the membrane really need to be anchored that tightly?
Well, the anchor isn't just holding the membrane itself. It's holding crucial biological machinery in place on that membrane.
Machinery.
Yeah. To understand why that matters, we have to talk about auxin.
Okay. The growth hormone.
Right. Auxin is the primary plant growth hormone. It's the master regulator that tells the plant where to grow roots, how to bend toward the light, and how to sense gravity.
It's the plant's internal GPS and steering wheel combined.
Exactly. But auxin doesn't just diffuse randomly like, food coloring in water. It has to be actively and directionally pumped through the plant tissue to create specific pools of hormone.
Which makes sense if it's directing growth.
This process is called polar auxin transport, or PA. And to pump that hormone, the plant uses specific molecular pumps located on the membrane. They're called P-ion proteins.
P-ion proteins. So these P-ion proteins are physically shoving the auxin hormone out of the cell in a specific direction.
Yes. For the plant to grow correctly, these pin proteins like P-ion1, P-ion2, they cannot just float anywhere on the membrane.
Right.
They must be clustered tightly in highly specific polar domains. Think of them as tiny concentrated patches on just the very bottom or the very top of the cell.
How small are these patches?
Incredibly small, only about 300 to 600 nanometers wide.
Here's where it gets really interesting. Think of these pin proteins as directional thrusters on a spaceship.
Ooh, I like that analogy.
Right. If those thrusters are locked tightly at the rear of the hull, the ship flies straight. But if the hull is slippery, and those thrusters just slide around freely, firing in random directions, the ship will just spin chaotically.
Exactly. The plant needs those pin proteins locked down, and that lockdown is completely dependent on the boron-GIPC tethering.
Oh, so if the tether breaks, the thrusters slide.
Yes. When the researchers used that PBA chemical cap to break the boron bridges to the cell wall, those tightly clustered PIN2 polar domains fell apart almost immediately. The proteins just diffused randomly all over the plasma membrane, exactly like those sliding thrusters you mentioned.
So without the cell wall anchor, the machinery just drifts.
It drifts, and the cell recognizes that something is terribly wrong. In many cases, when the pin proteins lost their anchor, the cell just panicked and swallowed the proteins back inside itself.
Like it absorbed them.
Yeah, a recycling process called endocytosis. The pumps were completely removed from the surface.
That completely redefines how we view a plant's anatomy.
Yeah.
I mean, the cell wall isn't just a passive, protective cardboard box keeping the environment out.
If we connect this to the bigger picture, it proves the cell wall is an active structural scaffold.
Yeah.
By tethering to the membrane, the wall physically restricts the lateral movement of these pin proteins. The rigid wall is actually dictating the plant's internal chemical signaling and directional growth.
It is a beautiful example of biophysics driving biology. But wait, there's a missing link here, isn't there?
What's that?
We know boron holds the GIPCs to the wall, and if that breaks, the pin proteins scatter. But how do the pin proteins actually hold onto the GIPCs in the first place?
Ah, right. How does the thruster attach to the hull?
Yeah, because we know the lipid is the anchor point. But a protein and a lipid are completely different types of molecules.
To answer that, they looked at the physical 3D structure of the PIN2 protein itself. Hidden inside its structure, specifically in the 6th transmembrane domain, the part that snakes back and forth through the membrane.
Right.
They found a conserved sphingolipid binding motif.
A motif being a specific pattern of amino acids.
Exactly. It's a specific sequence, TRLILIMVW. 9 amino acids that fold into a very specific pocket. Think of it like a custom-made lock that only fits one specific lipid key.
And that key is the GIPC.
Yes, the sphingolipid family, which includes our abundant GIPCs. To prove this physical interaction was real, they ran flotation and overlay assays.
How do those work?
Essentially, they mixed pin proteins and GIPC lipids together, spun them in a centrifuge, and proved that the proteins directly and exclusively bind to the GIPCs, ignoring all other control lipids.
Okay, wait, so the pin protein literally has a custom-built puzzle piece that exclusively fits into this GIPC lipid.
Yes, it does.
But biological systems usually have redundancies. Is this one specific lock and key mechanism really the only thing keeping the plant growing straight?
The precision and the necessity of this lock are staggering. The researchers used CRISPR-Cas9 gene editing to delete just 9 base pairs of DNA from the plant's genome.
Just 9 base pairs, that's tiny.
Tiny. That edit removed just 3 amino acids from that specific binding pocket in the pin protein. Essentially, they filed off 3 teeth from the lock.
Just 3 amino acids out of the entire massive protein structure. What happened to the plant?
It was devastating. That tiny dilution resulted in a mutant plant that had drastically reduced PIN2 proteins on its membrane. Its roots were severely stunted, and it suffered a total loss of gravitropism.
Meaning the roots literally didn't know which way was down.
They grew in chaotic loops. That phenotype phenocopies an AIR1 mutant, which is a known severe mutation.
Wow.
Deleting just 9 base pairs blinded an entire plant to gravity simply because a single protein could no longer shake hands with a specific lipid.
That is mind-blowing. The sheer scale of the causality there. A microscopic lock breaks, a protein drifts, a hormone scatters, and a root forgets how to find the center of the Earth.
And the connection relies on the lipid structure, too.
Oh, really?
Yeah, GIPCs have long tails made of VLCFAs, which are very long-chain fatty acids that plunge deep into the membrane.
Okay.
When researchers treated plants with an herbicide called metazaclor, which blocks the synthesis of these specific long tails, they saw the exact same chaotic unmooring of the P-ine proteins.
Because the anchor needs a deep root.
Exactly. What's even more profound is that this binding motif is conserved across almost all land plants.
So this isn't just some quirky feature of a single laboratory plant.
Not at all. This highlights that the GIPC-pine interaction is a foundational biophysical mechanism. It is quite literally the evolutionary adaptation that allowed ancient plants to conquer dry land and develop complex gravity-sensing root systems.
Because without this tether, plants would have basically remained formless aquatic blobs.
Pretty much.
Yeah. Okay, let's take a breath and do another quick recap. We've traced the molecular snaps, the boron bridges connecting the cell wall to the GIPC lipids, and the custom lock and key proteins holding the hormone pumps in place.
A very elegant system.
It is. Let's bring this all back to the farm, to the soil, to the farmer, looking at a withered crop that we started with. What does this entire cascade look like in the real world when a plant is actually starving for boron?
To connect the micro to the macro, the researchers studied Arabidopsis plants with a mutation in a gene called NIP51.
NIP51, okay.
This mutation breaks the plant's boron channel, meaning the plant suffers severe boron deficiency internally, even if the soil is fine.
Oh, interesting. So what happens to them?
The symptoms were textbook agricultural failure, exhausted root meristems, and a total failure to establish auxin maxima.
Those auxin maxima, those are the critical concentrated pools of hormone at the very tips of the roots that tell them to keep pushing downward into the soil.
Yes.
Yeah.
And the researchers visualized this using a DR5.GFP fluorescent reporter. It's a genetic trick that literally makes the pools of auxin glow green under a microscope.
Oh, that's cool. So you can literally see the hormone being there.
In a healthy plant, the root tip has a bright, glowing sweet spot of auxin right where it needs to be.
And in the mutants.
In the boron-starved mutants, that glowing sweet spot completely vanished. The hormone was just scattered diffusely throughout the tissue or gone entirely.
So the signal is just lost in the noise. Did they look at any actual commercial crops beyond the lab plants?
They did. They checked Brassica napus, which is rapeseed, a massive global agricultural crop.
Yeah, huge crop.
And they looked at the boron mutant. Under low boron stress, these plants develop bizarre protruding stigmas.
Wait, their flowers get deformed?
Yeah, their reproductive organs literally grow outward chaotically because the internal signaling is completely unmoored.
A total structural breakdown.
And the final definitive proof came when they used IPUT1 RNA eye lines to genetically knock out the plant's ability to synthesize GIPCs altogether.
Oh, wow. So they just removed the lipid anchors from the equation entirely.
Yes. And when they did that, the plants suffered the exact same severe dwarfism and root defects as plants starved of boron or quadruple pin mutants engineered to entirely lack the P-ine hormone pumps.
So what does this all mean? When you put all of that together, what it means for agriculture is just profound.
It really is.
When a farmer sees a withered boron-deficient crop, they aren't just seeing a generally sick plant struggling to metabolize. They are witnessing billions of microscopic physical anchors snapping simultaneously.
Snapping all at once.
The cellular membranes are literally pulling away from the cell walls, the directional thrusters are sliding all over the hull, and the plant's internal GPS has completely crashed.
That is the core synthesis of the source material. Defective polar auxin transport is the primary biological reason for boron deficiency symptoms in crops.
The 100-year mystery is solved.
It is. Boron is the essential linchpin of the cell wall plasma membrane continuum. It holds the lipids, which hold the proteins, which direct the hormones, which ultimately build the plant.
It is an incredible cascade of causality to think about. Just to wrap up here, we started with a dying crop in a field, zoomed into the cellular wall, discovered a tiny chemical bridge made of boron and saw how that biological Velcro allows the directional flow of growth hormones.
It's amazing.
It takes a trace element we barely think about to give a plant its shape and its ability to sense gravity.
This raises an important question, though. It forces you to rethink how biology utilizes the basic elements of the periodic table.
How so?
Well, if boron, a simple, incredibly basic element, plays this highly specific structural cross-linking role in plant cell walls, what other seemingly simple trace elements are doing heavy biophysical lifting that we haven't even discovered yet?
Oh, wow. Like in our own bodies.
Exactly. What seemingly random minerals in our own human diets are secretly doing crucial structural work.
That is exactly the thought to leave you with today. The next time you take a multivitamin or look at the trace minerals on a nutrition label, don't just think of them as vague nutrients floating around in your bloodstream.
They are structural.
Ponder the microscopic architecture of the natural world. What invisible chemical tethers are out there right now holding your own cellular machinery in place?